risk assessment for hard gelatin capsule cross-linking and dissolution method development. This is the topic for the video. I welcome all the viewers to this important video. Whenever we are working onto the hard gelatin capsules, whether you are going to fill the powder, granules or pellets or combination of formulations like pellets and tablets or other combination, that time there is a need to perform the risk assessment for hard gelatin capsule cross-linking, as it will affect the dissolution. Whenever there is a cross-linking in the gelatin, the dissolution will get hampered. The dissolution results will be on lower side. The drug will be trapped inside the gelatin shell. It will not get released, or variable results will be there. So a risk assessment is necessary, and if you found that there is a risk, then depending on the risk, dissolution method development to be done. We need to use certain enzymes so that the cross-linking can be broken down, and we can show that or demonstrate that the dissolution slowdown is because of the cross-linking. So it is also included in the USP monograph and general chapters. What is cross-linking? First we need to see what is cross-linking. It is cross-linking of the peptide in the gelatin, and because of that cross-linking or chemical modification in the gelatin, there is a formation of the pellicle, or there is a formation of a rubbery layer, and this pellicle is not soluble. That's why it slows down the dissolution. It also affects the disintegration, and through the formation of insoluble pellicle, it affects the dissolution. Then why we need to assess the risk? So risk assessment is important and required to ensure the consistent drug release, to meet the regulatory expectations, and to optimize the dissolution methods under stress conditions. If you don't do the risk assessment, and if cross-linking happens at storage conditions, then you will get surprise during the stability studies. So that time you will need the dissolution method with enzyme. So whenever you are working on the capsule formulation, do the risk assessment early during the method development and during the formulation development stage. Now see the causes of cross-linking. There are different causes, like intrinsic factors are there, like capsule shell components, APIs and excipients. Extrinsic factors are storage conditions, exposure to aldehydes or oxidants, capsule interaction with the drug, or interaction with some excipients or composition. Storage may affect the capsule shell and may induce cross-linking because of the heat, humidity and light. So capsule storage is very important, and we should protect the capsules from excessive heat, moisture and light. Regulatory concerns. So cross-linking capsules may fail in dissolution, and because of that the regulatory bodies like USFDA, EMA, ANVISA require the robust dissolution methods. So we can use enzyme to justify the cross-linking, and we can demonstrate that the slowdown in the dissolution or variation in the dissolution is because of the cross-linking, and it is not related to other formulation or processing problems. We need to do stress testing in the method development. Now, how you can induce the cross-linking or how you can induce the pellicle formation. So first is confirm the cross-linking susceptibility. Second is validate the enzyme strategy, enzyme use in the dissolution media, and then prove the method robustness. Then we need to validate and use that method. So purpose of inducing cross-linking deliberately. As we know that cross-linking is a time-dependent phenomena sometimes. So we need to induce the cross-linking to develop the method. Confirm cross-linking susceptibility, simulate real-world stress, then identify the vulnerable batches early. Induce the pellicle formation. Then validate enzyme strategy. Test the enzyme effectiveness to break the cross-linking. Evaluate the impact of surfactants like SLS. If your media is containing SLS, that time SLS may not be compatible with enzymes, or the enzyme may not work in presence of the SLS. So we need to test the enzyme effectiveness and we need to check the effect of surfactants. Then prove the method robustness, demonstrate method reliability under stress, and strengthen validation and the regulatory documentation. As our aim is to make the robust dissolution process, method should be validated, verified, and should be suitable. So by testing the cross-linking capsules, we can demonstrate the dissolution method reliability. Now see the methods to induce the cross-linking. How you can induce the cross-linking. So the reasons for cross-linking are there may be presence of aldehyde like formaldehyde. There may be some impurities. There may be some residual impurities in the blend or in the field material. There may be the cross-linking because of heat and humidity, sometimes light. So we need to deliberately prepare the batches so that cross-linking can happen. Now, first is formaldehyde vapor. Store the capsules in closed chambers with the formaldehyde vapors. Expose the capsules to excessive heat and moisture. It may be 40 degrees Celsius, 75% RH, or sometime you can stress the samples. Then exposure to the light and formaldehyde spiking in the fill material. You can add trace amount of formaldehyde to the formulation composition and then check whether the cross-linking is happening or not, because cross-linking many times happens because of the aldehyde. Tailor condition by capsule type, fill composition, and stress intensity. Sometimes you will not get stress cross-linking because of heat and humidity, but you can get by the formaldehyde. Then go for the risk identification and assessment. You can use tools like FMEA and fishbone diagram. Parameters like we can monitor the disintegration time, percent release, and physical shell integrity. You should observe the dissolution of the capsules and you should observe the pellicle formation. Enzyme strategy and media considerations. Enzyme use pepsin 0.1% in 0.1 normal HCl or pH 1.2. Sometimes pancreatin can be used if the dissolution media is basic. So depending on to the media pH, we can use the enzymes, and we should use the enzymes at such a concentration which are optimum for the dissolution. Also, we should refer the USP method. We should refer the USP chapters for enzyme concentration. Then surfactant conflict. Surfactants like SLS may denature the enzymes. So this needs to be evaluated. USP two-step strategy. So you can use the pre-treatment type of method. Step 1, pre-treatment with enzyme only medium for 15 minutes and then add surfactant containing medium to complete the dissolution, so that you can avoid the denaturation of the enzyme and you can get the required dissolution. Integration into method development. So apply cross-linking stress to placebo capsules and final formulations. Include enzyme pretreatment and compare the data. Justify approach in development reports and regulatory submissions. And also you need to finalize the enzyme level based on the media composition. If it is acidic, then pepsin is used. If it is basic, then pancreatin can be used. Nowadays, sometimes bromelain is also used if the pH is around 4 to 5. So we need to develop the method. Risk mitigation strategy. Insert capsule materials like HPMC capsules. You can check the dissolution with the formulation batches made with HPMC capsule shells in place of the hard gelatin capsule, because HPMC will not show the cross-linking. Avoid reactive excipients or aldehyde generating drugs. Optimize fill pH and moisture levels. Monitor the storage and distribution conditions. These are the risk mitigation strategies. And if you find that the risk is there, then develop the method with enzymes. Capsule cross-linking is a critical risk to the dissolution performance, and many formulations which are the hard gelatin capsule formulations show the cross-linking. Risk assessment enhances the product robustness, regulatory compliance, and patient safety, and it will reduce the problems or surprises in the stability studies, and it will save your time if you develop the dissolution method early during the product development. Enzyme-based dissolution must be scientifically validated and justified. So it is obvious that if we are using enzymes, we need to be very sure regarding the level, and its use should be justified. Thank you for watching the video. If you have any questions, you can mail me and you can comment. I will try to answer. I request you to watch my other videos related to hard gelatin capsule cross-linking. These are available onto the channel in the playlist, so that you will get the good information about this topic. Thank you.